So today I'll tell you about recent work on adaptation by large jumps. And just to set the stage, I'm going to tell you about the first case of adaptive evolution that was observed in nature. It's a classic case in all the textbooks. It's a case of evolution of a peppered moth in England. So this is the peppered moth. It's sitting on a tree. You can barely see it. But when industrialization started happening in England and the trees got dark, it got much easier for us and for the birds to see these peppered moths. And at the beginning, these moths were this color, but eventually they got darker. I'm not sure you can see. Eventually, this moth, the dark moth, became prevalent. And in the span of just maybe a decade, it became the dominant type in England. Actually, later on in the 1960s, 70s, the peppered moth morph shifted back. So this is an example of adaptive evolution. There was a genetic mutation that spread through the population, and we can now we know what the mutation is. It's a single mutation, one particular gene called cortex, and it provided about 50% benefit to the individuals that carry it. So how do we think in general about adaptation by mutation? And what's the current way of thinking about this kind of case, but also in general about how evolution has brought about all of the beautiful diversity and function in biological systems? So the kind of the standard way, and it's definitely the way that Darwin thinks about it, is that evolution proceeds through accumulation of a very large number of very small, in fact, mutations over a very long period of time. It's a slow and steady process. So it's something that would be hard for us to observe in real time because it occurs on sort of geological timescales. Historically, the other way of thinking about evolution was evolution by saltation, by big jumps, by single mutations that can bring about large changes. And so this is one example of such large mutation that has actually observed. It's a single mutation in, actually pair mutations in Drosophila that moved legs onto the place where antennae usually are. And so these mutations can occur, but people usually think that they're unlikely to be adaptive. But there were people, in particular Richard Goldschmidt, who proposed that this is how evolution in general happens. It happens through these jumps. But this notion was derived as the notion of evolution proceeding through the occurrence of this whole hopeful monsters. And the fact that many of you probably haven't heard of Richard Goldschmidt connotes that this view is not particularly popular these days. So why is the view that evolution will proceed through accumulation of very small mutations is prevalent? And it comes less from empirical data, but comes more from just basic theoretical assumptions. So if you think of evolution as optimization problem occurring in multiple dimensions, and you think of organisms having many dimensions, many parts that are all integrated together, kind of one way to analogize it is to think of trying to get a microscope into focus, and you have multiple knobs to turn. And if mutations turn multiple knobs at once, and the more knobs there are, the harder it is for you to put the microscope in the right place. And if you will do it, you will have to do it by very, very small adjustments. Big changes of all the knobs at once will not work. And people have built, in particular, Ari Fisher, and then others have worked on it's called geometric model of adaptation when you think of organism existing in some kind of multidimensional trait space. So let's say it's here, and the optimum is somewhere at the origin of that space, and mutations move you in that space. And if you go towards the optimum, you're advantageous. If you go away, you're maladaptive. And the higher the dimensionality of that sphere, the less likely a random vector is going to be pointing towards the center. The curvature increases. And as the curvature increases, you expect that the mutations that will be adapted, will be pointing in the right directions, will have to be small. It's so-called, the notion is cost of complexity in biology. So if we go back to our moth, we think of individual mutations affecting possibly many traits. Organisms are integrated, they'll be affecting many traits. And as such, the this is my figure showing that biology is complex, right? That's just there's a lot of complexity in there. Everything's integrated, and so the big changes are likely to have some deleterious side effects, and the only ones that will be advantageous will be small changes, right? That they happen to move the colony in the right direction and did not affect many other traits in a detrimental direction. So that is a view that's currently prevalent, and it's in many ways is supported by modern quantitative genetics and the so-called omnigenic models that suggest that every mutation affects every trait. So having said all of that, how is it possible then that we do see these adaptive changes? They happen in nature. There's one particular mutation has 50% advantage. It's not tiny. It did happen. So one possibility is that it's just weird one-off example. We don't see anything like that in other systems, but that's not the case. I'm just going to show you two, but let's say there are many case studies. For example, these case studies in sticklebacks, there are marine forms and then there are freshwater forms. The sticklebacks move to the lakes, and they repeatedly lose plating and lose this pelvic spine. In both cases, there's a single large effect mutation that generates this. In cancer, the mutations that kind of the driving oncogenic mutations tend to hit single mutations, let's say in the gene called KRAS or in RAS family that I'll talk about more in yeast and the system. That can have very large, tens of % benefits for the cell in terms of its ability to divide. So the individual mutations can cause large effects in many systems, and there are many other examples. So, of course, there could still be anecdotal because we do detect large changes more easily. So there's some question about that. But nonetheless, it's not impossible. So the question is, how can it work? And I think that the current thinking about it is that most likely it's because there is some ability of some mutations to be modular, to change only one trait and not others. And this way you get out of the problem of the cost of complexity. You don't move all the knobs at once. You just move some knobs, but then you can move them by a lot. All right. So that's kind of the basic theoretical sense of it. And so the question is, how do we move forward, and how do we start thinking about it? And one thing we need to do is to somehow build more of these maps from genotype, from mutation to phenotype to fitness, to try to really assess how do mutations move phenotypes in real organisms and how they affect fitness. And we need to build this genotype phenotype map, and hopefully we do it for a very large unbiased sample of mutations. Unfortunately, so far, the study of adaptation has been really limited by numbers and by sort of classic textbook examples, but they tend to be we expect them to be biased in some way. We don't know how to generalize from them. So we need to have representative samples of adaptive mutations. And so one way to get at it is to use microbial systems and study evolution in the lab, kind of experimental evolution systems. You can do it with E. coli, like Richard Lansky has done famously. But in our lab, we use yeast. So what's great about yeast is that they're microbial, but they're also they're eukaryotic. They have a sexual phase and an asexual phase, and we also know a lot about yeast. We know a lot about cell biology of yeast. They're also used in making of food or brewing of beer. So there's just a lot of knowledge, and so studying experimental evolution in yeast it piggybacks on a lot of knowledge of the system. And so that's what we decided to use in the lab to start asking questions, these basic evolutionary questions. So I'll tell you today about one experiment, or maybe a couple experiments, but we're going to use the same setup. We're going to be studying yeast growing in a 2-day cycle in a media that's limited by sugar. And in that condition, they go first through a period of fermentation where they just eating sugar and converting it to ethanol by glycolysis, and then once they are out of sugar, they start slowly metabolizing ethanol, and then they eventually go to starvation. In that particular experiment, we don't let them get to complete starvation, and we replenish the media, and they go through the system again. So, but there are two physiologically, metabolically, two distinct stages of fermentation and respiration. And so this is how the experiment goes. We have a flask that goes, the number of cells goes from 10 to the 8 to 10 to the 10 in 24 hours, in 48 hours. And we replenish the media, it goes again, goes again, it goes again. And during the time, adaptive mutations can happen. So in that system, there's no sex, it's just clonal lineages evolving. And we know that many adaptive mutations happen in that system, and they have this kind of start in different individual cells, but then those cells, if they have an advantage over the ancestral type, they start expanding, and eventually they start taking over the whole culture. So that's kind of the setup. In order for us to follow what happens, we need to follow individual lineages. So mutations will happen somewhere in the genome, but the way the system works is that we introduce into the genome a very large number of unique DNA barcodes, and then because there's no recombination in the system, a mutation that happens anywhere in the genome, if it gives an advantage and starts increasing in frequency in the population, it drags with them the particular piece of DNA. There's a very large diversity of this piece of DNA. So if we find that particular kind of a word increasing in frequency, we know that something must have dragged it to high frequency. And so we only sequence the region with this DNA sequence barcode, so DNA barcode, and use them to figure out the underlying dynamics in the system. So the whole thing was invented by a former postdoc at Stanford and Gavin Shenlock's lab, Sasha Levy, and all of the work, the analytical work was done by Jamie. I found this picture of them yesterday. They're two great scientists being absolutely terrible at pretending to be scientists. So I love that. So this is the data we got. I just really love this slide, one of my favorite. So what's showing here is the data on the frequency trajectories of these individual barcodes. So this is time in generations. So every transfer is about 8 generations, right? So for every 48 hours, we have 8 generations. So 1, 2, 3, 4, so forth. These individual lines are individual barcodes, and the red ones are the ones that increase in frequency more than you would expect by chance. And you see there's just a lot of red lines. They're all unique lineages, and some of them increase in frequency very fast. So this is a log scale, so straight line means exponential increase. And so they start increasing in frequency, kind of eventually dominating the culture. And in this system, we detected maybe 10,000 unique adaptive mutations that increase in frequency in parallel. So there's just a lot of action. So one, it was interesting to see that it's happening, but also it's a really beautiful system for generating a very large sample of adaptive events, because individual barcodes tell us that if mutation happened in the background with a unique barcode, it must be in a different mutation by origin. Therefore, we have a way to get a large number of adaptive mutations and study them. And the system works, you go at a particular moment in time, and then you just plate the cells, and at that moment, roughly 30% of the culture consists of adaptive lineages, but the individual adaptive lineages are all rare. So you just plate it, sequence individual colonies, and then you know that barcode is unique, and then you figure out which mutation it has. And it's present in this strain, and most of the time we just get a single mutation in the whole genome. We know what it is, and we have also the clones themselves, so we can study them. We can do whatever we want to do with them. So the one thing we can do with them is that we can, once we figure out what they are, we can pull them back together, and now we can remeasure their fitness in bulk. So let's say you take 500 individual clones. There are different mutations. We know what they are. You pool them, and you regrow them together in competition with the ancestor. So you can measure fitness of those 500 individual mutations recurrently, many times, in the media in which they evolved or in any other media. So it's a very good way to get a large number of adaptive mutations and then study their fitness effects across environments, as one of the things you want to do. So that's what I'll tell you about. So this work, just want to acknowledge, former research scientists in Gavin's lab, Barbara Dunn, and my former grad student, Sidney Venkataram, who have done all of this work. So I'm skipping a lot of the technical steps. At the end, you get all these clones, and you figure out what the mutations are, and they come kind of in a few classes. So one interesting class of mutations was the initial cell is haploid, just one copy of the genome. It turns out that having two copies of the genome is by itself adaptive in the system, so diploid yeast do better by substantial amount. There's some structural variants. We don't know what they do, but the majority of the mutations that were observed were in the key regulatory pathways that sense amount of sugar in the environment, and the cell decides whether to grow or not to grow. They're called RAS-PKA and TOR-CH9 pathway. Kind of a lot of abbreviations. I'll just tell you briefly what that is. But the individual dot here is an individual mutation. So there's kind of many recurring mutations in the same genes and the same pathway. And you can also measure their fitness. So they're all not tiny. Like, this is not tiny little things that Darwin is talking about. This individual mutation, let's say this one, gives 120% advantage over the ancestor. And these are not some extreme environments. This is just regular media of a lab strain grown for 48 hours. So just entirely reasonable conditions. I did not expect there to be such big jumps, and there's just a lot of them. So we're now in a position to start studying what they are, what they do. So this is, say, one glucose sensing pathway, and this is another pathway that is implicated a lot in mammalian cells, in aging, in other. So another interesting thing about this pathway is that they're entirely conserved in all eukaryotes. So, and part of my lab does studies of cancer, and it's the same exact pathway that we see in cancer. All right, so this is the pathway. There's a lot is going on, but there's a gene that's involved in sensing the amount of glucose. There's some signal transduction, eventually goes through this protein that can exist in two states, bound to GP or GDP, kind of flips back and forth. There are genes that regulate whether or not it's likely to switch from on or off stage, and eventually the whole thing goes into, kind of transduces to a kinase, and the kind of decision is made somehow to whether to grow or not to grow. So, and we have a lot of mutations in this pathway in different parts. I'll be talking about some genes. So let's say this gene GPP2 here, PDE2, IR1. So they sit in different parts of this pathway, but mutations in them, and most of them are loss of function mutation, tend to be adaptive in this particular context. Okay, so the whole question for me was, can we learn something about these large effect adaptive mutations? So how can we ask questions about whether or not they're modular or not, how many traits they affect or not? And this is a signaling pathway, so maybe they just do one thing. There will be one trait that changes, and they all kind of do the same thing. Let's see whether it's true. So in other ways, can we figure out how many different traits these adaptive mutations affect, and is it true that they are modular? So, and this work was PhD work, of two really great PhD students, Grant Kinsler, who's now doing a postdoc at UPenn in cancer biology, and Yuping Li, who just started her own lab in Basel in Biocentrum. All right, so how do we study it? So I'll tell you two ways. So the first one, I think, very straightforward. So again, looking at what we've done, we've been evolving this population in this particular cycle when they go through fermentation respiration. Fermentation respiration are different physiologically and metabolically different stages. So can we figure out whether or not they gain fitness advantage either in fermentation stage or in respiration stage, what happened to them. We're just going to decompose it, and we can do it by measuring their fitness in just growing in 1 day. So we just grow them in 1 day and just measure how well they do. And then we measure them in 2 days, and the difference in fitness between 2 days and 1 day is roughly, we're doing something a little bit more sophisticated, but the basic idea is that the extra gain in fitness of growing in 2 days must be due to respiration compared to its fitness in 1 day. So I'm not showing you the data, but just measure things in 1 day, in 2 days, and subtract one from the other. Okay, so I'm going to be plotting here kind of the advantages mutations have in giving you in fitness advantage in fermentation, measured in fermentation per hour, and respiration. So, but basically the easiest way to think of it is x-axis is how much gain you have for the first 24 hours during fermentation, how much gain you have by just sitting in ethanol for 24 hours, and after that. And so there are several ways of thinking of it. Well, one of them is that secretly, even though the textbooks say otherwise, but secretly fermentation and respiration really are the same thing. And if whatever you gain in fermentation, you'll get the same amount in respiration. So they will be the same. The other way is that there's a strong trade-off, right? So that somehow if you improve fermentation, you'll hurt respiration and vice versa. You're already sitting on some kind of a Pareto front. Another way is that it's entirely modular. So some mutations will, the evolutionary condition has both fermentation and respiration. So maybe some mutation help you in fermentation, some mutation in respiration. They're all individually modular, but together you can move both of them. So kind of same related by trade-off and modular. At least these are the hypotheses that I had when we started thinking of it. And when we did it, it turned out to be completely different. It was like none of those. And in fact, the majority of the mutations were coherent. So they improved both fermentation and respiration together. There were mutations that improved one, and there are mutations that improved the other at the cost so you can improve fermentation at the cost of respiration, you can improve respiration without improving fermentation. But the biggest ones and the ones in the RESPK pathway were here. They got the biggest advantage because they improved both at once, and it looked like they were entirely capable of moving phenotype, improving in both fermentation and respiration, again, as two entirely distinct phases. So there's a little bit of evidence of kind of Pareto front that there were no mutations that were able to improve fermentation respiration to the maximum possible observed degree. There was nothing here, and we know that we would have been able to see those mutations because they would have been really adaptive. So there's a little bit, you can go all the way diagonally, but you can go diagonally pretty far, right? And then the question becomes, what's going on there? Is it what yeasts do? It's just easy for them to move respiration fermentation. Again, there really are kind of similar-ish in some way, or something else is going on. And so we decided to do the kind of obvious next experiment and ask, okay, this was one step. What happens in the second step? And then it's like, let's take a few places and start asking whether it's going to continue the same way or not. So we took some, instead of taking the ancestor who was here, we kind of redid the experiment, but started with the ancestors close to the boundary, and kind of started thinking again, what are the possibilities? One is this, we call it not modular, but pleiotropy. Pleiotropy is the opposite of modularity. So pleiotropic adaptation is common, and you will continue going at 45 degrees. And the other possibility is that it's rare, and maybe we already got to some kind of Pareto front. Now you finally have the trade-off, and you're going to go kind of 45 degrees, but minus 45 degrees, like the opposite. And if you're going to evolve, you might be able to evolve only kind of along one axis. So we did it, and we took 5 different starting points, redone this experiment, and the bottom line is that, so this is the IR1. This is one of the kind of negative regulators of RAS. the first step, something like 86, almost 90% of mutations were improving both. But when we look at R1 missense, the second step from that starting point, the majority of them just went up. And in fact, maybe the majority of them improved respiration, but hurt fermentation a little bit, right? So it's changed drastically. If we look at another one, R1 nonsense, it was even stronger. So they went up, right? But not diagonally. The next one, and the next one. And so in every case, at least particularly in these five examples, you no longer go 45 degrees. You are going straight up. And the genes, instead of RAS-PK pathway, they tend to be genes that are related to respiration, as you would expect. And a lot of them are also in some signaling pathways, just like with RAS-PK, but now related to mitochondrial function and respiration. Okay, makes sense. Interesting, if we ask, like, well, is that it? You just cannot improve fermentation anymore. We just, by chance, got a few cases where there were multiple adaptive mutants in there, and we found in generally that, let's say in this particular case, the first mutation, the common one, goes up, but then the second one improves fermentation, and you start kind of overall going in a 45-degree angle, but now it takes at least 2 steps rather than 1 step. So the first one, you could just go diagonally, and now you have to go kind of up and over. It just takes 2 steps to move at 45 degrees. So it looks like, at least from this data, it looks like in this example, the first step was pleiotropic, but coherently so, and then you moved to more modular behavior, although this modular behavior is also probably, if we decompose respiration to more parts, there'll probably a lot of things to also kind of, there's also pleiotropy there. Okay, so this is the kind of the main result of the first part of the talk. So we have a lot of coherent pleiotropy at the beginning, and then it shifts to modularity. So there was something special about that stage that allowed this coherent pleiotropy. So if we ask the question, well, do you have to be modular? No. So the adaptive mutations are not necessarily modular. And in fact, in some cases, you can have kind of coherent pleiotropy, but it's coherent and in some way low dimensional. So you shift something that can at once move multiple traits, but you're not moving them randomly. They're moving them in somehow sensible way. You're improving both of them. All right, so this is great, and kind of, I think we got some insight from it, but it was kind of non-generic, which is a problem in that we needed to know a lot about yeast, about physiology, and we could only look at this particular 2 performances. So we took overall fitness and decomposed into 2 performances. But the question is, can we do something a little bit more general and ask a question about sort of dimensionality of adaptation and how many traits are you affecting if you have large effect mutations? And again, we want to build this genotype to phenotype to fitness maps. So I'll tell you it's a large project in the lab. I'll just tell you about kind of the first, our attempts and how we're trying to do it in a way that does not force us to know a lot about the phenotype and a lot about the actual physiology of the organism, just trying to do it as abstractly as possible, but maybe eventually get to the facts of the matter. So the reason we want to avoid knowing too much about what the phenotypes are and what the cell does is because it's a complete awful mess. Like there's just a lot of things are happening, and if I know that mutation changed my fitness, let's say improved my fitness in general, it's very hard for me to know at what level of organization it happened, which phenotype to measure. Everything is co-varying and correlated, and it's just hard to marginalize one particular effect over others. It's very hard to assign causality. So instead of kind of Carrie Geiler in my lab called it genotype to phenotype to phenotype to phenotype to phenotype to phenotype to fitness map. It's very difficult. So then it was like, well, maybe we can get some insight into it by measuring no phenotype whatsoever, but instead doing the following. Let's say we have a mutation IR1, this one nonsense mutation in this one particular gene, and we know that it's improved fitness. what if we just measure its fitness? We're not learning anything about the actual, actual parts in between genotype and fitness. We're just measuring fitness, but we're going to measure fitness in a bunch of different conditions. And I'm going to measure fitness in a bunch of different conditions that are close by. Just I'm going to jiggle my environment, and I'll measure fitness really well. And so I'm learning how the fitness changes as environments change. And then I'm going to imagine that there are some orthogonal phenotypes in there that the mutation affects. And I imagine that these phenotypes interact with some environmental variables. And there is some kind of dimensionality k between the number of phenotypes that affects and the number of the environmental variables. And my fitness is just this one particular instantiation of this thinking is just linear. Just we multiply the two vectors, and we get a scalar of our fitness. So this is for one mutant, but now I can do it for another mutant, GBB2. And I'm going to do it in the same exact conditions, and I'll get these profiles. And so every particular condition has its own particular vector of effects, and every mutant affects the same traits. And so now I have, I have finite number of mutants, I have finite number of conditions, but the total amount of data I get is large. And so then the question is, can I decompose all of that into something lower dimensional? And in this particular case, we're going to use this entirely standard linear algebra, singular value decomposition, kind of similar to PCA. So we're going to just say, what is the dimensionality of underlying effects? How much do we need to know in order to predict the fitness of all of these mutants that we have? So we have like 400 mutants. How much information do I need to assign, predict the fitness for every mutant in this condition, every mutant in my collection in the conditions I'm studying? So maybe I need just one dimension, maybe I need two dimensions, maybe three dimensions. Let's just see what we actually get. So the nice thing about it is that, again, another question, one, we have no idea which phenotypes to measure, but the other thing is we have no idea which conditions to use, right? Because we have no idea, again, despite the fact that it's Saccharomyces cerevisiae, everybody's supposed to know everything about it. We have no idea what yeast actually, quote unquote, feel about the environment. What do they care about, right? I don't know. I think that I'm changing sugar. I'm changing something about its glycolysis, but maybe what matters to them at that moment is osmotic pressure or something else. I really have no idea how to do it. But the nice thing about this entirely abstract approach is we'll just do a lot of random stuff that's easy to do. And they'll just see, and we'll try to do it so it's not always the same thing. And so at the end, as long as our perturbations are sufficiently uncorrelated, maybe it will give us enough way of shaking the system that we'll learn how many things are really moving inside the cell. So here we're using 400 mutants in 45 different conditions, and some of them change the amount of time they spend in fermentation, some different sugars, some salt, some drugs, just different things. And it was a lot of work. So Grant Newping and Sandeep already mentioned, and Carrie Geiler-Samarodi was a postdoc in the lab, and now she's her own lab in Arizona State. And so they pooled their efforts together, and they made this large measurement. And I'll tell you kind of briefly what we found. I'm skipping, again, a lot of details, but I'm happy to, in Q&A, answer questions. So what I'm showing you are the fitnesses of some of the key mutants, and they're going to be arranged on this axis, where these first ones is their measurement in different batches in the condition we evolved them in, right? So this is kind of where we started. And then I'm going to, the conditions are ranked in order in which in general, they induce differences in fitness across the mutants. So this condition is most like to this condition, but not because I believe it to be so, but because the fitnesses of the mutants were most similar between this condition and M3. And this condition gave me most different fitnesses. So as you go on the x-axis, the fitnesses will change more dramatically. So this is the first example. So this is the most adaptive mutant in our condition, R1 nonsense. It had, and I'm pulling all of the mutants in that class together. So it's quite adaptive on M3, it stays adaptive, but then when you get to very high salt, they die. And so that's one thing that you already see, that these mutations are not just generically adaptive, they're kind of conditionally adaptive in a particular condition. And again, it tells us like, these are large effect things. These are not tiny little things. You can be 100% better than the ancestor, or you can be dead, right? The ancestor is perfectly fine here, but this guy dies. So if we look at another one, diploid, it turns out to be generalist. They're always good. They're always better than haploids. And it's kind of interesting why. I actually don't know why. No, we haven't figured it out. But it's interesting that yeast generally live as diploids out there. OK, so this is GPB2. There was less adaptive mutation, but it changes its fitness. It also gets worse in salt, but not as much as iron nonsense. PDE2 also is doing its thing. So GPB2 and PDE2 are very similar close by, but eventually they start doing different things. So these genes are different. They're doing different things. And there is some kind of interesting pattern of fitness across the environments. So this is all the data. And so again, we have 400 mutants, and we have all these profiles. And then what we do is we split them into subtle perturbations and stored perturbations. Subtle perturbations, basically, they're the changes of the fitnesses that were within a standard deviation of the M3. So they're very similar to their evolving condition. And the reason we want to do just subtle things is that we don't want to completely change. We want to understand what they were doing in the evolving condition. That was our idea. So we don't want to go too far from it. And for kind of in a kind of applied math kind of way, there is a hope that if you do only subtle perturbations, the whole system should behave in a more linear way. You can kind of do Taylor series approximation there, and things should linearize. So the fact that we're using linear models is not insane. So that was kind of our notion. The minus is that you have to measure things really well, because otherwise you'll just be dealing with noise rather than with actual biological signal. And just for brevity, although I have slides to kind of go more into detail if people want to go more into detail in Q&A, the main result that we got is that at least locally, you don't need that many numbers. The dimensionality is fairly low. So we start with average fitness of these mutants, and that is the best predictor, but it's kind of boring, because all these conditions were subtle perturbations, so they basically look like evolving condition. So the fact that the average does well is not interesting. But the other dimensions are kind of G by E dimensions, the interaction between the genotype and the environment. And then we really just need something like 4 dimensions to explain everything. So, and again, I don't have the slides here, but I have them, after the acknowledgements, if people are interested. We do really well in subtle perturbations. We can use this model to predict things quite well. They kind of weirdly work even if you go far away from the evolving conditions, but that's for another talk. But interesting that all of these mutants can be, their fitness effects can be placed in a fairly simple space. So maybe 3-4 dimensions is all you need, right? So that also connotes this notion that there is some low dimensionality in the whole thing, that these mutants doing something that's not there, even though they're different and they could have different effects far away, but locally, near the evolving condition, they all behave in a fairly simple way. They're not moving too many levers. So, so I'm coming to the end, just to say that these kind of experiments, I think, are telling us that adaptive mutations don't need to be truly modular, in a sense that they need to affect only one trait at a time, and they cannot affect too many traits. But in fact, at least in these cases, we see quite a bit of coherent and low-dimensional pleiotropy, where you can move multiple things at once, but in a sensible direction, such that overall you're adaptive. And just kind of going through the whole thing, what I showed you that we start, just to kind of recap and also try to think like what it could possibly mean. So we have a system to get a large number of adaptive mutants. We know what they are. They tend to come from this signaling pathway. And we find that they move multiple traits at once, and then they're low dimensional. But I think that one thing that I keep coming back to, and this is where at least this is a hypothesis that we have right now, is that in most of these interesting mutants are in these very conserved, high leverage regulatory pathways. They're not just kind of at the edges of what the organism does. They're kind of at the core. This is a pathway, again, that's conserved across all eukaryotes. And the thinking then is that, well, maybe in general, these hopeful monsters, this large effect, large jumps, maybe in general, the reason they're possible is that they hijack regulation. So in this case, it's a regulatory gene. In both of these, the pelvic spine, lateral structure, these are genes involved in regulation. In cancer, it's all, not all, but most of them are regulatory modules that, the receptors and the main signaling molecules, including this RAS pathway. So maybe what's happening is that the regulation is already pre-wired in such a way that it has to move multiple parts of the organism in a sensible way. That's why these regulatory pathways exist. That's what they do. They can change the cellular state from quiescence to really rapid growth. And when you go from one to the other, in cell biology textbooks, it's very unfortunate, but there's like blah, 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 growth. And it's like, I have no idea what growth is, because growth seems like a lot. You have to put everything together just the right way. You have all this stoichiometry. You need to completely rewire the whole cell. But the cells are capable of doing it. And so if you want to adapt rapidly, you probably want to do something about those levers that already exist. It's sort of like if you want your car to go faster, you probably don't want to go with a screwdriver into the engine. You probably want to put a brake on the gas pedal. And it's going to be easier. And the gas pedals are already pre-wired to allow your car to go faster without ruining everything at once. And so kind of going back to the original theoretical reason for why we think adaptation must proceed through small steps in this kind of geometrical models or in the analogy with the microscope, I think the problem with those analogies is sort of kind of the general fable of a spherical cow, or call it a spherical horse, is that organisms are just not spheres. And they already have parts that are wired together. Of course, they're complicated, and you can easily break it, right? So that's why most mutations are deleterious. But also, they're wired in a very sensible way, and they can build themselves. And there's development, and there's regulation. And both of those parts allow adaptation to use the systems that are already coherently pleiotropic, and not just random throwing vectors in this entirely symmetrical space the way theoretical biologists have been thinking about it. So that's what I want to say, is that real organisms have low-dimensional, high-leverage, coherently pleiotropic regulatory paths that enable large adaptive jumps. And that's what we see in cancer, that we see in these very rapid adaptations to changing environments. One question is, it's not clear whether or not this will generalize to a longer-term evolution compared from a short-term. Maybe eventually you need to do something different, and maybe evolution of novel structures is going to be completely different. So maybe we're not learning in these experiments about evolution over a longer period of time. And I think it's an interesting question to ask. I don't know yet how to ask it, but at least if you're thinking about adaptation to change of the environment on a short time scale, thinking that adaptation cannot proceed to large jumps is both theoretically and empirically incorrect. So just want to thank people who contribute to this work. I already acknowledged Yuping, Grant, and Carey. Just want to acknowledge Gavin Sherlock, who's a professor of genetics at Stanford and was the inspiration for all of the work we've done in the system. And his lab kind of developed the barcoding system. And in general, he's been a great collaborator. And just want to thank my lab, and thank you for listening.
Okay, well, just two quick questions. One is, what is the measure of fitness in your yeast experiments? I wasn't sure what you meant by fitness.
By fitness, I mean the advantage over the ancestors. So how fast does the lineage increase in frequency.
So it's like the numbers. You're counting how many, or you're measuring how many.
I'm looking at the slope of change of the numbers of cells.
Oh, okay, the numbers of cells. And the other question is, this thing about large adaptive jumps, is this related in any way to this sort of punctuated equilibrium idea that's been out there for?
So punctuated equilibrium, for people who know, it's a pattern in paleontological data that the species tend to look very similar for a long period of time, and then they change. It might be, but my gut feeling, that's not what's happening, that organisms are constantly evolving, and then sometime ecology changes and they move, and they start seeing that that can exist. But I don't know. But it's a fact that punctuated equilibrium can happen, might relate to this kind of ideas. But I don't know how to relate the
Might
two.
be on different scales.
Might be different.
Yeah,
and also,
exactly.
punctuated on paleontological scales is different.
Right, a different scale. Yeah.
I have no idea.
I see.
So many interesting things there. Many of the mutations that you talked about and identified in that screen came from the RAS nutrient sensing pathway in yeast. And it looked like, from what you showed, you got some of the components of that pathway, but certainly not all of the components in that. And I wonder if there's information to be gained by going to that pathway and now saying, why didn't we get mutations in these other components? Is it because they have too small of an effect, too big of an effect, not enough of a 45-degree angle effect? Have you tried any experiments like that?
So it's a great question. We haven't done exactly that experiment. We are, but we're gearing up to do it. We're using these high throughput CRISPR approaches where you can CRISPR in a pool and get on the order of 10,000 variants and with barcoding and measure their fitness. So and we've done it for natural variation in the RAS pathway. It just turns out to be very interesting. So because one of the complaints about this is maybe experimental evolution is completely irrelevant to nature, but we find that it's not the case. But one of the things is do kind of deep mutational scanning of all of the components, and I think it's fascinating. Just to finish, one I said is unbiased sample of mutations. Of course, it's biased because we need to have sampled them. They needed to have been frequent enough and adaptive enough. So it's not entirely unbiased, but maybe we can do something more unbiased, like Tim is suggesting. I don't know. You want to?
You often don't hit this side. Okay.
Okay, I'm on. A couple of questions. So one, I was fascinated by the group of mutations that you got and how they affected fermentation versus respiration. I wondered if there was, if the switch is actually, part of why you had the sort of limit in the first case, and then you could, have mutations that could then allow you to accomplish that switch more quickly. And maybe I missed it, but I would have guessed those would be, the respiration components or something that would allow you to switch into metabolic respiration. That was one question. The second question was, I wondered if the duplication of the genome is influencing some of what you're able to obtain in this approach versus, say, for example, a multi-copy approach where you might be able to get dominant effects and bypass some of the problems with redundancy in the yeast genome.
So great question. So the first one, I don't know what limits it. I think that this RAS-PK pathway is probably wired to move fermentation respiration coherently because it's not the first time that yeast ate sugar and then ended up with ethanol and ate ethanol, right? So they must have already regulatory systems that do both. But it's interesting, you can all go as far in 45-degree and it requires additional steps. Maybe, we're only sampling some mutational space. Maybe there are some mutations that are less likely to happen. We just haven't seen them. They're possible. We're using haploid here, so because they evolve faster. So the diploid happens, it's a kind of a separate case. But it's true that because we start with haploids, Structural variation is going to be undersampled. And so we're also doing things in diploids, like say something like loss of a gene might be lethal in that case, whereas if you go from diploid 2 copies to 1 copy might be advantageous. So spectrum mutations will be different.
Right. Actually, in that case, I was referring not to the haploid versus diploid, but to the actual genome.
Oh, that
duplication.
happened. Yes, that's
Yeah.
very interesting. We don't see anything related to there. So for the yeast went through ancestral, 100 million years ago, the whole genome duplication, and there's just a lot of redundancy in the genome.
Exactly.
But we don't see that. But it's an interesting question. Maybe that's affecting it.
You hijacked my mind. I'm curious what your thoughts are on epigenetics, and I had a real-world experience when I was a contractor at Foster Farms in California, where they were trying to get the chickens to lay in the nests. But they were laying in the ground. They picked them up at night and they put them in the nest in little cubicles. And the ones that laid eggs from the first generation on, laid them back in the cubicles. How do you make sense of that?
Well, I think there are epigenetic changes. I think there is transgenerational inheritance in animals. Doesn't seem to be very strong compared to plants. There's a lot. It's interesting. I think it could have a real effect as organisms can, let's say, not die over the big change, and then they can adapt. So that also can happen. We are trying to, as much as we can, stay away from it. From epigenetics, we're trying to go to genetics just because we want to have more repeatable things. But maybe it's to our detriment that we're staying away from it. Maybe it might have a real life effects.
Thank you for your talk. You mentioned that the first mutation tended to affect both the respiration and the fermentation fitness, but then the second mutation was not like that. Why the change?
So at least the hypothesis, maybe I didn't state it cleanly enough, I think the RAS-PKA pathway is unique. It is the thing that can move you this way. Once you've used it up, now you're done. Now you have to do other things. Now you can get there, but you need to take another, let's say, mitochondrial thing and then something else. And then you might go like this. And we're doing the experiment. We're trying to see which way it's going to go. Is it going to tend to go one or the other? Are you going to be a generalist? Are you going to become a specialist? But I think these pre-wired pathways, they're finite. There's just not infinite number of them. So use one up, use one up. The same in cancer. You hit MAP kinase pathway, and the next thing you need to hit is PI3 kinase pathway, and then something else, and something else, and something else.
Thanks for the talk. So how do you explore the perturbation space for a multicellular organism? How do you explore that perturbation? So you tested, did one perturbation test the fitness? How do you explore that for a multicellular organism?
How do I explore the space?
Yes, so because the signaling, there are much more other signals that may relate to their function. So this is space.
I think it's a really fascinating question. So right now, we, of course, in this experiment, we're bounded by the mutants that we have, and they were all evolved in a particular case. So the next step is to start with a much larger set of mutants. And one thing we're doing right now is we're taking a collection of natural isolates, and we're just analyzing the data. We just take a collection of natural isolates. They're all barcoded as well, so you can pull them together. You grow them in a insane number of conditions, and then you're trying to say what is the space that they themselves have, and they have been evolving in nature in multiple conditions. So hopefully they have explored much of the space. The other one is more kind of Tim's approach. We know some of these pathways. We can go, bash RAS pathway, TORC pathway, HOG pathway, whatever pathway we want, that we know supposedly what they do, and just see what is the pattern of pleiotropy we'll get there. That's a great question.
Let's take one more question.
Hi, this is a meta question. So you have for a lot of species like your yeast, they have an advantage if they're able to mutate because they have some flexibility. But in the very same medium are the mitochondrial bodies, and they have a huge need to not mutate, otherwise almost certainly kill the organism. So it seems to me that there's something profoundly bigger than just like, the nature of mutation, that there has to be a reason why mutation is valued for the continuing existence.
I think you're touching. So there's kind of maybe putting in other words, you're putting in the kind of question of evolution evolvability, like what makes organisms evolvable, and I think it's a fascinating question. It's kind of people trying not to work on it too much. Somehow it's just difficult to measure. Because you need to have something that varies in terms of evolvability. An organism already are whatever they are, but it's maybe not impossible. But I guess what I would kind of, at least my gut feeling is that if it's true that regulation and adaptation are two sides of the same coin, then it's almost you get it kind of for free. Because if you want to survive, you need to be able to sense the environment and respond in sensible ways, and then you need to create a system that is low dimensional but moves a lot of things at once. But then by doing so, you also allow paths of adaptation to happen. So my hope is that maybe something will come out of this, but we are far away from saying anything beyond just And you see, as I started waving my hands a lot, it means that I'm just less certain, and I'm waving them a lot.
I think it's time to give you a rest. And to
Thank you.
our speakers.